In chapter 3, we're going to be moving on to look at a variety of topics that are associated with cells. We'll start out looking at the sort of origins of life and some of the history behind some of what was going on with that. And then we'll switch gears and start looking at the features that we find in different cells. And we'll take a look at something that you can find in all cells and then go through the characteristics of the different parts of prokaryotic and eukaryotic cells in what I like to call the tour of the cell. And we'll end up at the end of this with looking at something known as the endosymbiotic theory. And just sort of as a reminder, when you look at something that is known as a theory, in science, this is different from what a lot of people use for the normal use of the word theory. When somebody says, I have a theory about that, what they're really saying is, I have a hypothesis. I have an educated guess about that. And in science, that hypothesis is tested through experimentation, and then the results from that are used to verify whether or not your educated guess or hypothesis was correct. And if enough people can agree that your hypothesis is correct, that's when we move on to say that your educated guess is a theory. And so if you look at this and think about this in terms of the endosymbiotic theory, that means that this is something where somebody made a guess and the experimental data that has been collected seems to bear out their theory. And if we look sort of at history, we can find that there's really two models that people came up with for the origins of life. One of those models or thought is what's known as abiogenesis, and the other one is known as biogenesis. And if we look at abiogenesis first, this is really the thought that life can arise without having pre-existing life. You can think of it as what's known as spontaneous generation, that these living organisms are just going to appear from non-living things. So if you think about this over here, you've got this pile of all these citrus fruits. Well, where did that mold come? It just arose without pre-existing life there. Other people looked at, you can see the maggots appearing in dead animals that you find out there. Or if you think about a pond that dries up, well, when it rains, that dirt must change into these living organisms because that pond is all of a sudden going to have frogs and fish and whatnot appearing there. And so it's kind of an interesting idea when you look at this, and probably one of my favorite examples of abiogenesis that people came up with, and this would probably be about 1,000 years ago, is they found something out there in actually Ireland called the goose barnacle, and they said, I have a theory about this, that the barnacle goose is actually coming from this goose barnacle. So this is kind of an interesting twist on life is coming from pre-existing life without descending with similarities. So this thing sort of evolved into something else. So kind of an interesting little twist on that idea. But people even did experiments on this, and there is a famous I guess you could call him a researcher out there, Jean Baptiste von Helmont, who actually proposed recipes for the creation of different kinds of animals that were out there. And his recipe for the creation of mice was to take some dirty clothes, put some wheat in there, and let it sit for about a month, and all of a sudden mice would appear. And so if you think about this, he was doing scientific experiments. He did this experiment, he made his observation, and he came to the conclusion based on his observations. So he was, in a way, following this idea of scientific method and drawing conclusions based on your experimental evidence that is out there. Nowadays, we accept this idea of what's known as biogenesis. And so if you look at this and think about abiogenesis, bio is life and genesis is creation. And so abiogenesis is the creation of life without previous life. The A is the without. But here, life is created from similar kinds of life. And we can really summarize this and say that living organisms are descending from similar organisms, or you can look at it as cells produce cells over here. And this is the commonly accepted thing, that we use. And again, this is based on a lot of experimental evidence. When we look at this phylogenetic tree, we know that there are links between our prokaryotic cells that are out there, and eukaryotic cells appear to have descended from the archaea ancestor, and all of these things descend from a common ancestor that is out there. And so this is based on scientific experiments and a lot of people getting together and looking at those results and drawing conclusions based on the observations and the results of those experiments. And this idea of abiogenesis has really been around for since before the Greeks were doing their sort of philosophical science. These ancient beliefs over time started to become tested by people that were out there. And really, the first major experiment to challenge the idea of spontaneous generation occurred in the mid 1600s by this guy, Francesco Redi. And he basically challenged the idea that people were saying maggots are arising spontaneously from rotting meat that was out there. And he performed what we would consider a relatively simple kind of experiment. Put an open container with meat out there, and you see maggots appearing. So there's your spontaneous generation. He also put out a sealed container and found that there were no maggots being formed in there. And then a third thing over here is a lot of people said, well, in the sealed container, you aren't allowing air to get in there. And remember, they had this idea of miasma, that there was some force in the air out there that was causing diseases. So why not a force in the air that's responsible for life? And so he used a cloth, conveniently enough, that you could find in Italy, that allowed air to pass in and out of this container here. And what he found is that you did not see the appearance of your maggots on the meat in that container right there, but you did see some of these maggots arising on top of the little gauze covering right there. And because he was doing this in Italy, and again, think about the time, the mid 1600s, the Catholic Church said, well, this supports the belief that living things must originate from parents. And that sort of played into that idea of what was going on in the church, and a lot of the stories from the Bible that your parents are out there, but sometimes you can have a spontaneous birth of a child out there that must be special as a result of this. So when we look at this idea, this is starting to show that the macroscopic um, or big versions of life don't arise spontaneously, that there's something else going on here. And in this case, you've got your flies that must be coming in direct contact with your meat right there. But if you think about it, okay, we don't have maggots on our meat, but is something else going to appear on that meat? And yeah, go back and think about the citrus that I showed before, molds and other kinds of things that are going to appear. So people started to wonder about what's going on at the microscopic level with these things. And to do experiments on that, people would use basically broth experiments. So think like gravy right here. And when we look at this, you can have a clear gravy that you let sit out, and over time it's going to start to turn cloudy. And there is your evidence of bacterial growth. And notice the time. This is when microscopes were becoming prevalent, and so people were putting this broth underneath the microscope and able to see these little animalcules moving around in there that weren't there when you had your original broth that you let sit out for a few days. And so there is this belief that if you boiled stuff, you could kill all living things that were in there. And Needham did some experiments where he took some broth that had cells in it potentially and boiled it, and that killed off the cells. And then he put a little plug in this guy over here, and what he found was that over time, this broth became cloudy again. And if you looked at it under the microscope, you would see your little animalcules swimming around or moving around inside of there. And so this belief really perpetuated, or this experiment perpetuated the belief in spontaneous generation, and it was able to persist for longer. And a lot of people started to scrutinize these experiments and look to see what was going on. But many of the scientists of the day agreed with his experiment and his conclusion that microbial life seem to be able to spontaneously arise in your broth right there. But of course, people started to do more and more experiments with this, and another Italian guy named Spallanzani did some more broth experiments. He modified Needham's experiment. So you take the same kind of setup here, but his big difference is he sealed the container before he actually did the boiling. And when he looked at his ones, he did not see those cells appearing. As long as this flask is sealed, it remained clear. So that eliminated the spontaneous growth of bacteria cells there until you opened them back up. And Needham argued with Spallanzani about this and said, well, there is some sort of life force in the air that must have been destroyed by this process of your boiling here. And other people said, well, if we can open these things up, maybe microbes are floating around in the air and that's landing in there, and that's where we're getting the growth of these things. As long as they're sealed, things cannot get in there. So people went back and forth doing these kinds of experiments by boiling broth and looking at the results of that. But it really wasn't until 100 years later that Pasteur came along and finally settled that question of spontaneous generation. And he took a flask but made one big change. He made what's called a swan neck on his flask. So imagine just taking the top of this thing and stretching it out really far and then allowing it to bend so that you get this little curve that looks kind of like the neck of a swan right there. And this allows air to get in and out of it. But when you have contaminants floating around in the air outside, when they come into this bend, they basically are trapped inside of that bend. So this is going to allow your gases, the vital force in the air, to get in here, but prevent basically solid material from being able to enter into this flask. And when Pasteur did these same boiling experiments, this little guy would remain clear for months on end. And that is a really good argument that you're not going to have this microbial growth because the cells that are floating around in the air aren't able to contaminate it. But again, people are going to argue about that. And they said, well, maybe your boiling disrupted the life force that is in this, which really isn't a good argument because people had seen that boiling could still allow the growth of some of these things out there. And so to refute that, Pasteur said, okay, I'm going to take my swan neck flask, boil it, kill off the microorganisms that are in there, and then let it sit and show there is no growth. But over time, you can break off the neck of the flask, and that allows these cells that are floating around out there to land in your sterile broth, and you get the growth there. So this is showing that the contamination is what is causing the growth inside of there. And so this really simple experiment was able to overturn literally thousands of years of thought about the origins of life. And so now we have this theory, the cell theory of life, that cells come from other cells and that biogenesis is what we should be looking at when we're looking at the creation of life. But if you go all the way back to the origins, remember, where did that first cell come from? Well, that is something that is a whole different class out there. I did want to point out, just to put things in perspective with the timeline over here, when you look at this, the Greeks proposing that miasma theory, you're talking 2,000 years ago. And here we can see the origins of the microscope with Robert Hooke in England, and von Leeuwenhoek over here was the first person to really see these single-celled organisms that are out there. And so these observations with the microscope led people to start to propose this idea of the cell theory of life in the 1850s that Pasteur came along and showed with his swan neck flask experiments that the cell theory seemed to be true. We can look at the germ theory for microbiologists that are out there. And there's an Austrian guy named Semmelweis who worked in a hospital, and he made an observation that a lot of women who came into the hospital to give birth were picking up what's known as ward fever, and it turns out to be a bacterial infection. And during the time there, the medical doctors that were in training would be doing things like autopsies, And when a woman came into the hospital to give birth, they would all run over there, and whoever got there first would be her doctor and could help in the childbirth process. Well, in the process here, they weren't washing their hands. And so Semmelweis made this observation that maybe these people were carrying something on their hands that was responsible for these disease. And so he had his residents and interns basically wash their hands in what's known as a chlorinated lime solution. So think bleach, wash your hands in bleach water. And he actually showed by looking at the number of women who died of ward fever that when his students were washing their hands, the number of deaths dropped off drastically. Unfortunately for him, people didn't believe that washing your hands was actually a good thing. And he toured around Europe trying to get people to believe this. And because people didn't believe him, he got depressed and eventually was put into an insane asylum where he, it turns out, picked up a bacterial infection that resulted in his death, ironically. There is another guy over here that had some better public relations named Lister, and Listerine was named in honor of him. He didn't come up with it, but he came up with this idea that maybe we can use a variety of different kinds of chemicals to treat the area of a surgical wound or your surgical equipment and decrease the amount of infections that might be occurring. So basically, same idea, wash your hands, clean things off, and that will decrease the amount of infections that occur as a result of a surgical procedure. So like I said, better public relations, so he did not go crazy and had some mouthwash named in honor of him. When we look over here at John Snow, he was an Englishman who basically came up with what we refer to as epidemiology now. So what the Center for Disease Control is doing with the COVID virus pandemic is basically tracking where these things are occurring. And if you look at this, back in the 1850s, he tracked a cholera outbreak that was occurring in London and basically sat down with a map and was able to look at it and say, you know what, all of this surrounding area, we see cholera going on in people, and cholera is basically massive amounts of diarrhea as a result of a bacterial infection. And so he went to the center of the circle that he drew on the map where the outbreak was, and he found a water pump there. And the story goes is that he took the handle off the water pump and the cholera outbreak dropped off in London because people were no longer drinking that contaminated water there. So you can see a variety of just kind of interesting things here. This is by no means a definitive list of microbiologists, but just some of the more interesting kinds of things that are out there. And the last two over here, Robert Koch, is a guy, and I talked a little bit about him in some of the labs. His lab came up with the petri dishes, and they isolated the microbes that are out there. So the streaks that we're doing in lab to isolate pure cultures, and what they were able to show is I can isolate this bacteria from a diseased animal, and when I administer that isolated bacteria to a healthy animal, it develops the disease. And when I look in that healthy animal that did not have the microbe, all of a sudden they have this new microbe. And so he was basically able, he and his lab workers were able to define this bacteria causes this disease. And they showed it first with anthrax and went on to show it with a whole variety of different kinds of bacterial isolates that they made. And I added one more thing in here because if you notice, this is all male heavy. There are women who are doing some work in microbiology, and one of the more famous ones, Florence Nightingale over here, she's the one who basically was a hippie in London, and she ran away and went to help out in the Crimean War that the British Empire was fighting on the continent. And based on her observations as a nurse there, she said, you know what, it's probably not a good idea to have a toilet trench right next to where we are doing surgery. And so she set up various standards for nursing and sanitation during this time frame here that basically changed how we looked at contamination that was going on, and was able to use statistics to show that if we make these changes, you're going to see some benefit at the patient level there. So think about this idea of the history of microbiology, the idea that cells arise from other cells is what really drove a lot of the science that's going on here, because that's what led to the germ theory, that germs are what's responsible for causing diseases, not bad air, not that witch with the evil eye that I talked about in the first chapter.